This is Dr. Mitch Robinson, and this video is on carbohydrate digestion and transport. In this video, I'm going to discuss the digestion of dietary carbohydrates, and we'll look at the enzymes needed to convert the large molecules that we eat into small molecules that can be absorbed into the bloodstream. And we're going to look at transport molecules that are needed to move the monosaccharides across the cell membranes into the cells. Okay, so let's start by looking at the molecules that make up carbohydrates in our diet, and this includes monosaccharides and disaccharides. But the largest portion of the carbohydrate is primarily starch, and this is either amylose, which is a polymer of glucose molecules linked by alpha-1,4 bonds, and amylopectin, which has alpha-1,4 branches in addition to the alpha-1,4 chains. And we've looked at these molecules before, and you should be familiar with their structure from the Guts module on carbohydrates. Now I should mention that cellulose is a carbohydrate that is an important component of fiber, and fiber is an essential nutrient in our diet. But this molecule cannot be broken down by humans because we do not have the digestive enzyme that can hydrolyze the beta-1,4 bond of cellulose. So we cannot hydrolyze cellulose, and that's what makes it indigestible and actually a good component of our diet for that reason. Okay, so let's examine the steps in the digestion of starch, and this begins in the mouth with lingual or salivary amylase, also known as alpha amylase. And this enzyme is an endoglycosidase since it hydrolyzes the alpha-1,4 bond on the interior of the carbohydrate chain. Endoglycosidases work on residues. Exoglycosidases work at the end of the chain, but this enzyme will cleave the 1,4 bond at various portions inside the polymer. Salivary amylase is inactivated by the acidity of the stomach, and after the stomach contents move on to the intestines, the acidity is reduced by bicarbonate, and then pancreatic amylase is secreted into the intestine and takes over and continues the digestion of starch in a manner similar to the lingual amylase. Pancreatic amylase hydrolyzes the 1,4 bonds again in the same way. So eventually the starch you eat is converted to short chains called dextrins. Now the digestion of carbohydrates depends on enzymes that are present in the membranes of the intestinal epithelium, and this is known as a brush border membrane. The first of these enzymes is alpha-glucosidase or glucoamylase. This is an exoglycosidase since it hydrolyzes from the non-reducing end of the chain. So this enzyme will cleave the chain from the end sequentially. The end product of starch digestion is now mainly disaccharides of glucose, either maltose, alpha-1,4 glucose, or isomaltose, which is alpha-1,6 glucose. Okay, so now we have enzymes that can hydrolyze these disaccharides. Maltase will hydrolyze maltose to 2 molecules of glucose. And I should mention that glucoamylase will also hydrolyze maltose. Isomaltase is required to hydrolyze the alpha-1,6 bond of isomaltase. In addition to starch, we have disaccharides in the diet that need to be digested since we can only absorb monosaccharides into the bloodstream. Sucrase is a component of the brush border membrane that will hydrolyze the 1,2 bond of sucrose to yield fructose and glucose. We need the enzyme lactase to hydrolyze the molecule lactose, and so lactase hydrolyzes the beta-1,4 bond of lactose to yield galactose and glucose. It's worth mentioning that sucrase and isomaltase are both enzymes, 2 enzymes that are located on a single protein. And some individuals have a deficiency in the gene that codes for this protein, and if that occurs, then you have a deficiency both of sucrase activity and isomaltase activity, and we'll talk more about that in class. Now, as you know, lactase is needed in all young mammals because it's needed to hydrolyze lactose in milk. But many adults lose the ability to hydrolyze lactose. It's a natural thing for the enzyme to decline with age. So in many of us who have an insufficiency of lactase, we have difficulty digesting milk and dairy products, or we have difficulty digesting the lactose that's in milk and dairy products. And this results in digestive problems, gas, bloating, diarrhea, when the lactose makes its way down into the large intestines and gets worked on by bacteria down there. Okay, now let's look at the transport of carbohydrates into cells. Glucose and other monosaccharides are polar molecules, and they can't cross the hydrophobic part of the cell membrane. So we need a transport protein that can bind these molecules and provide a channel so they can move across the cell membrane. First, let's examine the transport of glucose into cells, and then we'll identify transporters that move the other monosaccharides across the cell membrane. In most tissues, glucose is transported by facilitated diffusion. That is, it's transported along a concentration gradient. There are a family of proteins that act as glucose transporters by facilitated diffusion. They're known as the GLUT transporters. There are a family of genes that code for these proteins, and the genes are known as solute carrier family 2. So remember, solute carrier family 2 genes encode for GLUT transporter proteins. There are 12 members of the GLUT family of proteins that have been identified so far, and they all code for a single polypeptide chain, and that chain spans the membrane 12 times, and it forms a channel across the membrane that allows glucose and other monosaccharides to transport specifically across the membrane. Now, all of these GLUT members have different properties, and they're adapted to particular tissues where they're located. They're called glucose transporters, but some of them bind and transport other monosaccharides as well. Now, on page 6 of your handout is a table with the properties of the 5 glucose transporters, GLUT1 through 5. And these are the most common, the most well-characterized members, and the most important ones. These are the ones we'll discuss. And these are very important to glucose homeostasis. They're important to understanding glucose metabolism in general and to understanding disorders of glucose metabolism, such as diabetes. GLUT1 is found in most all tissues, and it's known as the basal glucose transporter. It's responsible for the low level of glucose transport that's needed to keep all cells alive. GLUT2 is found in the intestine, pancreatic beta cell, kidney, and liver. It is a high capacity and low affinity transporter. That is, it works only when the concentration is fairly high on one side of the membrane, but it has a high capacity. So this kinetic property allows glucose to move across the membrane in either direction, depending on which side has a higher glucose concentration. It's therefore known as a bidirectional glucose transporter. GLUT2, this property of GLUT2 is important in the liver because the liver must take up glucose after a meal when the concentration of blood is high, and it releases glucose into the blood when the level of glucose in the blood is low. GLUT2 is present in the beta cell in the pancreas, and this allows the beta cell to act as a glucose sensor and, depending on the level of glucose in the blood, release insulin at higher glucose levels. GLUT2 is also present in the intestinal epithelial cells, and all 3 monosaccharides, glucose, galactose, and fructose, are transported from the intestinal mucosal cell into the portal circulation by GLUT2. GLUT3 is found primarily in neurons, and it transports glucose into nerve tissue. GLUT4 is found in adipose tissue and striated muscle, including cardiac and skeletal muscle. The GLUT4 responds to the level of insulin in the blood. So as the insulin increases in the blood, the level of GLUT4 in the cell membrane increases, and so there's a greater transport of glucose into tissues, including adipose tissue and muscle tissue. GLUT5 is found in the kidney and intestine, and it actually acts as a fructose transporter. So all the members of the GLUT family again act by facilitated diffusion, meaning that glucose is transported along a concentration gradient. But glucose can also be transported against a concentration gradient by the sodium glucose transporter. This is a co-transport mechanism. This co-transporter binds to one molecule of sodium and one molecule of glucose and transports both molecules together across the membrane. The transport can occur against a glucose gradient because the sodium gradient is always much higher on the outside of the cell. So sodium is actually piggybacking off of the glucose gradient to be transported against its concentration gradient. Now the sodium gradient is maintained by an ATP-dependent sodium-potassium transporter. So the energy for moving glucose into the cell across the concentration gradient really comes from the ATP that's used to pump sodium out of the cell and create a strong sodium concentration gradient. So let's look at the transport of glucose across the intestinal epithelial cell to see how these transporters work together. And this is on page 4 of your handout. And this shows an intestinal epithelial cell with the glucose being transported from the intestinal lumen into the epithelial cell and across the cell into the bloodstream. And again, sodium is being pumped out of the cell, and the sodium concentration is being used by the sodium glucose transporter to allow glucose to be pumped from the intestinal lumen into the epithelial cell. Now GLUT2 is on the basolateral side of the cell, and this allows glucose to pass from the cell into the bloodstream. Now you may have noticed that in this diagram there are two sodium molecules shown being transported with each glucose molecule. And I should point out that there are different isoforms of the sodium glucose transporter, just as there are different isoforms of the GLUT transporters. And the different isoforms of the sodium glucose transporters transport different numbers of sodium and glucose molecules. So the ratio of glucose to sodium varies with the different sodium glucose transporter isoforms. Also note that these are generally referred to as SGLT, abbreviation for sodium glucose transporter. And also should be pointed out that these transporters are very important in the kidney as well as in the intestine. In the kidney, the SGLT isoforms are very important in glucose reabsorption. Okay, so that completes the video on number 4 on carbohydrate digestion and transport.